All right, today we are diving into a really clever discovery about how some of the most resilient and aggressive cancers manage to survive. It turns out they've figured out a stunning trick when they should be starving to death. As you can see, they've basically built their own internal recycling plant. Let's get into it. So, here's the big question that was really stumping researchers. You see, tumors are incredibly harsh environments. They're often cut off from the body's main nutrient supply. And yet, some cancer cells don't just survive in there, they actually thrive. So, how are they doing it? I mean, what are they eating when the pantry is completely bare? Well, the story really begins with this tiny but unbelievably important piece of machinery inside our cells. It's a protein that's supposed to act like a switch for cell growth. But in a huge number of cancers, that switch is broken. And that protein is called KRAS. The easiest way to think about it is like a simple light switch. In a healthy cell, it flicks on to signal, hey, it's time to grow and divide. And then, when the job's done, it reliably flicks off. It's a totally fundamental part of how our bodies keep everything in order. But in cancer, a mutation happens, and that switch breaks. It gets jammed permanently in the on position. Suddenly, the cell has completely lost its off signal. Now it's just stuck with this relentless, nonstop command, grow, divide, grow, divide. And that is what drives the out-of-control growth we see in tumors. And you know, this isn't some rare, obscure problem. This one single broken KRAS switch is found in a staggering 25 to 30% of all human cancers. That makes it one of the most common and dangerous mutations out there, especially in cancers of the colon, lung, and pancreas. But just knowing this switch was broken, well, that wasn't the whole story. Researchers started noticing something really strange. Not all cancers with this broken switch were behaving the same way, and that led them to a fascinating survival mystery. So to figure this out, scientists put these cancer cells through a major stress test. In the lab, they created an environment that was meant to mimic the inside of a real tumor by drastically cutting off their main food source, glucose. They were essentially trying to starve them out. And this is where it gets really interesting. A lot of the mutant cells did struggle or just stopped growing, which is what you'd expect. But two specific versions of this mutation, they're called G12D and G12V. They were incredibly tough. They just kept on going. They survived the starvation. Which, of course, brings us right back to that key question. If you take away their number one fuel source and they still survive, then what on earth are they eating? The answer, it turns out, is both brilliant and pretty sinister. You see, they weren't finding new food from the outside. They were making it themselves. They had completely rewired their own internal metabolism to turn their cellular waste into high-energy fuel. And the key player in this whole biological heist is a protein called FOXO1. Now, normally, FOXO1 is one of the good guys. It actually helps shut cells down during periods of stress. But in these specific KRAS mutants, the cancer hijacks FOXO1 and basically forces it to do its dirty work. So here's how that recycling process works. First, the hijacked FOXO1 protein goes and activates a gene called GLUL. You can think of that as flipping the main power switch to turn on the recycling plant. Then, that GLUL gene produces a special enzyme that does the real work. It grabs toxic ammonia, which is just waste floating around inside the cell, and it chemically converts it into glutamine, a super valuable, high-energy fuel that powers the cancer's growth. So, that's the crucial point. This cancer literally turns its own toxic trash into life-sustaining treasure. This incredible metabolic rewiring is exactly how it survives in the nutrient-poor environment of a tumor, when other, less adapted cells would just perish. But, and this is the really good news, this incredible survival mechanism, this unique dependency on its own recycling plant, also creates a massive vulnerability. Because if we know exactly how it's feeding itself, well, we can try to shut it down. Because the cancer is now so incredibly reliant on this little glutamine fuel loop, it absolutely cannot live without it. Its greatest strength has become its greatest weakness, and that gives researchers a very specific target to attack. So they came up with this two-pronged attack they called the double blockade. They used two different inhibitors to hit the system from both sides. The first one, called MSO, blocks the enzyme that produces the glutamine fuel. That cuts off the supply. The second one, CB839, blocks the enzyme that uses the fuel, which cuts off the demand.
It's a really comprehensive attack on the entire recycling loop.
And the results? Well, they were remarkable. As you can see on this chart, on their own, each drug had a little bit of an effect. But when they were used together, whoa, the effect wasn't just added together, it was multiplied. It's something called a synergistic effect, where the combination is just way more powerful than the sum of its parts. I mean, look at that dramatic drop in the third bar. The double blockade was devastatingly effective. They had successfully starved the starving cancer. Okay, this is obviously a huge step forward. But as with all good science, it's really crucial to understand where this discovery fits in the bigger picture. So, are we talking about a cure that's ready tomorrow? The short answer is no, not yet. This is a brilliant foundational discovery for sure, but it is so important to be clear about the scientific context here before anyone gets ahead of themselves. So here are the key things to remember. All this research was done in vitro. That means in lab dishes, not in actual people. The specific drugs they used, especially MSO, might be too toxic for clinical use right now. So what this work does is identify a powerful new therapeutic target. It is not an immediate cure. A lot more research is needed to translate this amazing insight into a safe and effective treatment for patients. But the real takeaway here is this. This is exactly why this kind of deep quantitative biology is so powerful. By meticulously mapping out precisely how a specific cancer mutation works and survives, we can uncover its hidden weaknesses. It gives us a blueprint for designing smarter, more targeted therapies down the road. And that really just leaves us with a fascinating final thought. If these very common cancers are hiding this secret recycling plant in plain sight, it just begs the question, what other secret weaknesses are out there just waiting for us to find them?